Hi everyone, welcome to this M2D2 talk. Our speaker today is Surendranath Bhagat. He's a senior scientist at Redesign Science, and before that, he was a postdoctoral researcher at the University of Pennsylvania, working at the interface of Markov state modeling and molecular simulation. He also holds a PhD from Lund University in Sweden, where his research was focused on understanding protein ligand binding and exploring conformational dynamics of biomolecules using enhanced sampling and molecular dynamic simulations. Now he's fully working at the interface of molecular simulation and machine learning in the context of drug discovery. Thank you so much, Bakat, for accepting our invitation to present your work here today, and looking forward to the talk.
Thank you, Prudencio. Thank you for the invite, and thanks everyone for joining. Hopefully you can all hear me well. The title of the talk is Accelerating Cryptic Pocket Discovery using Alphafold and Markov state modeling. So I'm going to try to make a story of why cryptic pockets are important in drug discovery context and how we can sample them effectively using a combination of Alphafold and Markov state models. So we have to go back and start seeing what's the big picture in drug discovery. In drug discovery, people usually target a pocket which is called orthosteric pocket or a classical binding pocket, and it ends up being a big drug discovery efforts by many companies or in academic settings. But cryptic pocket always provide an alternative strategy for drug discovery. It can help target a pocket which was not existent before in the protein, but now it pops up during simulation. And if you target that, you have a better way to optimize the drug molecule. It can also help design selective inhibitors. So if selectivity is a big problem in kinases, aspartic proteases, and in other targets, if you target a cryptic pocket, because you're targeting completely new area within protein, that selectivity is not a big issue because that kind of transient opening does not happen in all the other subtypes. It also provides a strategy to address the drug resistance problem, which is a prevalent case in antibiotic. So if you target like a beta-lactamase in a typical pocket, you will always get a drug resistance within multiple generations of drug discovery. But if a transient pocket opens up and you target the transient pocket, which is called a cryptic pocket, it tries to address that drug resistance problem. So it has many facets of interesting application in drug discovery. However, sampling of cryptic pocket is very difficult because cryptic pocket opening is a very slow motion. And here I'm showing a few examples of how the cryptic pocket can open up in protein. So one of the examples is the side chain flipping, which you can see in the figure left bottom, where the side chain flips, the beta hairpin moves, and a cryptic pocket opens up. The another option can be a secondary structure motion, which you can see in the right side of the picture, where there is a drug binds and a loop part completely shifted, and there is now a big pocket which can be accessible and can be accessed by small molecules. It can also be a part of a tertiary structure shift by a combination of multiple substructures that can move at the same time. So all these different kinds of motion happen, but it's very difficult to capture because all these motions are in a very slow timescale regime, so in the microsecond to millisecond time range, which is very difficult to capture from molecular simulations. And there has been strategies to capture long timescale events, such as enhanced sampling, such as machine learning combined with enhanced sampling approaches, but it's always difficult to get kinetics out of those approaches. So we wanted to address not only capturing the motion, but try to understand and get an idea of the kinetics associated with this kind of cryptic pocket opening. In this case, we are using Plasmepsin II, which is the antimalarial protein, as our test system. And you can see from figure 1, the gray one is the orthosteric pocket And when this tryptophan 41 flips, it's part of a hydrophobic pack. When it flips, the beta hairpin opens up and the cryptic pocket becomes accessible. So it's quite a big change, and one can see by doing a pocket volume analysis, when the pocket opens up from traditional to cryptic pocket, the pocket volume increases, and it gives us the opportunity to target that pocket by different small molecules. So that's what the exciting things comes. Now one can make if we can sample this kind of pocket, we can make a better drugs. So the approach which we took is slightly different. In traditional ways, we usually see the simulation from PDB structures, and we try to get some kind of understanding by performing long simulations or enhanced sampling, such as metadynamics, to get an idea of the landscape of the protein. But all of these simulations usually start from one defined structure. Usually, Alphafold also gives one typical defined structure. But if we play with these hyperparameters, which I'm showing on the right side of the image, Alphafold managed to generate a diverse structural ensemble rather than one structure, which has different side chain motions, as you can see in the figure here. And if we seed simulation from this kind of diverse structural ensemble and combine the simulations using Markov state model, we can easily go back and forth between an open and a closed state. So like cryptic pocket open and a cryptic pocket closed state. And that will give us an idea of how we can get the kinetics out of the simulation, which is a big challenge. So in this case, we can see that we have sampled multiple conformational states of this tryptophan in Plasmepsin II, also the tyrosine, and we will see how we launching simulation from this kind of diverse ensemble will give us a better idea. Moving forward, I think this picture better captures how the sampling of Alphafold happens. So I'm projecting a free energy surface, which is coming from enhanced sampling, such as metadynamics, which try to access all the possible areas. And the black dots are the snapshots from Alphafold. And you can see Alphafold managed to sample intermediate states, which is in, if you see my cursor here, that's the intermediate state. It's also sampled open state. It has a bunch of structures in the closed state and a bunch of structures from high energy states. So that kind of the ensemble we are talking about. And next, what we did is we ran multiple small independent simulations from these all black points. And we used Markov state model, which is a way to get kinetics from MD simulation to get more information about this open to close transition. Yes, Prudentia.
Yes. So I'm wondering, how do you like when Alphafold give you many different structures, how do you know without, is there a way to know without kind of doing the MD, like which one are like different low energy state compared to the high energy one? Like, is there any way to do
that? From a structure ensemble, you mean?
Yes.
Yeah, so from a structure ensemble, it's difficult to know which motion is a slow motion. So that's why we get an idea. We start the simulation from the ensemble, but when we analyze, we can use Markov state model to understand which is a slow motion, which is a fast motion, which we will see which I will describe in the next slide.
Okay. Thank you.
So here's the whole picture here. Now we have seeded the simulation from these structures. In comparison, we also seeded the simulation from the APO in the same length of simulation. And we can see these two very stark difference in sampling. So by using Markov state model, we can get a transition rate going from closed to open to address your previous point. Now we know it's a slow event. And in the APO seeded simulation in the same time scale, we have never sampled this much conformation. So this is a key point here that starting from this kind of diverse ensemble, followed by molecular dynamics simulation and Markov state modeling, we can manage to sample this flow event, which is not possible if we just launch a simulation from only apo structure or only a bound structure. Then we never get this much structured sampling diversity. And we also compare to the different approach to see if we are in simulation. It's always interesting to compare with different approaches. We also use the tryptophan chi1, chi2 angle as CVs in metadynamics and projected a derivated free energy surface. And here we are comparing the output from our Alphafold seeded MSM approach versus metadynamics. And you can see the free energy surfaces look exactly similar, except the barriers are slightly overestimated in metadynamics, which is quite well expected because it's changing the underlying potential energy surface by addition of bias. So it always have a high barrier. But in MSM approach, there is no change of potential energy surface using artificial bias. So it's statistically better to get a kinetics from Markov state modeling approach. So this gives us a proof that we are not sampling something artificial, and from two different approaches, we are sampling a similar kind of free energy surface. Can we extend this approach to other systems? So we try to extend to multiple proteins here. There are a few targets like beta-lactamase for antibiotic design. There's a bunch of influenza target, multiple different targets here. This red line, which is a 1.2 angstrom cutoff, this is the line where we have experimental hollow structures of cryptic pocket open. And we can see in many cases, we have sampled a percentage of structures which can be a cryptic pocket open state, starting from our Alphafold seeded approach. And a few difficult cases, we have not managed to sample that, which we expect. But the good part is that in most cases, we have the Alphafold managed to sample this kind of cryptic pocket open states, which gives us a better chance of understanding kinetics by combining it with Markov state modeling and MD simulation. And here is a picture on the right side, which was a slightly difficult cryptic pocket opening. You can see it combines a sidechain flipping with a loop motion, which is more difficult than only sidechain flipping. And it manages to sample that conformation as well. So the bottom line of the story is we can extend this approach to many diverse protein ensembles to have better sampling of allosteric pocket opening or cryptic pocket opening, which can be targeted by small molecules or other biologics. Next, we focused on, can we use this idea to understand an activation mechanism in a very critical drug target? This is a BAX, which is a activation apoptotic activator, and there is this blue part of the BAX, which we call alpha 9. It usually opens up and binds to the protein, binds to the lipid interface here, as you can see from this activation mechanism. On the right side, I have put only NMR structure, only one NMR structure that is available in PDB for the BAX, where you can see the alpha 9 sits on the helix region. It does not open up. So we wanted to see, can we use simulation to understand this activation mechanism? Because till date, there is no PDB evidence of how the activated alpha 9 will look. So in order to address that question, we use the same strategy. And here I'm plotting different conformations coming from Alphafold. And you can see it manages to sample inactive state, and mostly inactive state, a bunch of intermediate states where the alpha 9 dislodges, and a few active state where it completely comes off the hinge, the helix groove. So that's our starting point to run simulations. So we can already expect that the simulations will do better than just launching simulation from an inactive state. So here's the simulation, and I'm plotting the first two principal components, and the principal components are just the motion of the alpha 9 unit, which is now in the blue. As you can see, we clearly separated three basins here. One of the basin corresponds to the inactive state where the this alpha-9 unit, which is shown in blue, sits on the top of the other helices. The intermediate state, right, the part of the alpha-9 opens up, and the activated state, where it completely extends out in the solution, which will eventually bind to a lipid to activate a downstream or upstream pathway. So yeah, that ends my presentation. And just wanted to point out that we are working in some cool applications of this strategy in context of kinases and other disease varieties where we feel like understanding cryptic pocket opening and allostery will be a big part in designing selective inhibitors. So we'll be very happy to take any questions or any doubts regarding this.
Thank you so much, Bakat, for the presentation. Was wondering, can you maybe kind of elaborate a bit on how much domain knowledge versus goes into kind of this work? Like, is it the kind of, for someone who's trying to use simply ML to figure out where is the cryptic pocket? Let's say I have a new protein, I use the Alphafold to generate like a symbol of confirmation. How do I go from there? Like, I'm not an ML expert and I don't know as much as you about like all these different like I don't know as much about the protein itself. How do I go about it?
Yeah, that's a great question. So we can give the example of in the BACS. In BACS, where we didn't know much about what to look for. So if you generate a diverse conformational ensemble from Alphafold and you run small simulations, like I'm saying, for each of the snapshots, you run like 10 nanosecond of MD simulation, then we can try to train a machine learning model which will capture which are the important features that contributes to the conformational dynamics of the protein. So that's a very unbiased way without human intuition to understand which features are important for this structural change. But if you want more data, you can go by the Markov state modeling way. If you don't want to get more data, but you wanted to understand which features are important for particular conformational change, we can try to train a time-lagged autoencoder, which will try to capture important features from the simulation.
Okay, thank you. There's a few questions in the chat. From E. Malik, will you say changing around AlphaFold parameter is a form of research? And if so, how much of the dimensional space do you estimate you have covered?
So we have covered the extreme dimensional space here. So what I mean by extreme is that we have, as you can see, the inactive state, we have the active state, but most of the samples stays within a ground state, which is kind of inactive state in this case. But it also depends on how much diversity of different frequencies have within PDB. So if you have more diversity, it will try to capture, the search will be bigger. So it will try to get more coverage. I think that's the initial trend which we have seen within this kind of approach, but tuning the parameters. Does that answer the question?
Yes, that covers it. I was just wondering, it's like if it's also there's a limitations just because of Alphafold's parameters, like would there be things outside of those parameters that potentially are available to you but immediately unavailable?
Yeah, we can tune that. We are working on tuning a part of Alphafold scaffold to be more suited, but that's very customized. But on the other hand, the Google, there is the open source implementation of Alphafold, and which I highlighted few parameters here. And I think if you play around with these parameters, and there is the way to search the different sequence aligned space, right? Like there's MMSQ way of doing it. There's the Google has a default called jackhammer, which is finds more sequences than the default. So there has to be some kind of so if you check out these parameters, and I believe in most cases which we have seen, we have generated multiple like diverse ensembles for different proteins. But as I mentioned, it is somehow training on a database. So if the database already exists a bunch of diverse ensemble for a different homologs, it might find a better starting structure.
Okay, thank you.
Thank you.
Question from Ari. You say you are using a NMR PDB structure. Is it because they are so much more accurate than the other options? Can you elaborate on that?
So in the BACS case, we only have a PDB structure. In PDB, there is NMR solution structure for inactive BACS. That's what it has. So so when launching simulation, I'm not using the NMR ensemble to launch simulations. I'm just trying to give an idea that that's what we have. In Plasmepsin, we have crystal structures available. So in most cases, we have crystal structures available. Is there anything more in that question?
I don't think so, because it makes sense. From Sasha, how do you make sure the Cryptic Pocket found in Alphafold plus MSMS is relevant to drug discovery? If a pocket is not always available, would that affect drug binding efficiency?
Yeah, so we are currently working on a few such inhibitors, which we have seen very good activity in biochemical assay. I cannot go more in detail on them. And one of them is on plasmepsin, a different plasmepsin subtype, which we are working on, where there was no existing cryptic pockets, and we managed to sample that. And once we target them by small molecule, did an inhibition assay, the results were in the low nanomolar, and that's a very good starting point to optimize. And usually in the early days of drug discovery, it's a very good story. So for a few systems, we have this protocol, and we managed to get output and do like a real small molecule drug discovery. And just to add on, after publishing this paper, there was a different group which did a small molecule, like with a more medicinal chemistry approach, targeting cryptic pocket, and they reported a crystal structure with their small molecule, and they managed to sample the same cryptic pocket. So that's also another validation that the pocket exists in biology.
Perfect. Any thought on Rosetta Alphafold versus Rosetta Fold versus CSM Fold? Why initially did you choose to use Alphafold?
Yeah, so we didn't have the Rosetta Fold open source code base available to us when we started the project, neither the ESM Fold, but we believe all of these fold approaches can be tuned to generate the similar kind of ensemble. But because we started with Alphafold, we stayed with the same kind of code base. So no special preference on any kind of fold approaches, but eventually all will be converged towards, if you tune the hyperparameters, they will manage to generate the similar ensemble.
Awesome. Is the MSMS only supervised training, or it can be used in self-supervised or even a native learning setting?
So in MSM, it's just counting how many times it goes back and forth between states. So if I see here, it is just counting number of times it goes back and forth between open and closed. So you just give it like what is your feature of choice, and it will do that. But in a broader sense, if you have small amount of MD data from MD simulation, you can train a supervised algorithm to find out, for example, if you have 10 times, 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 10 ti to find out which features gonna be more dominating. And that has been done by training a supervised machine learning algorithm like a support vector machine, which can train on different features to pick which features which features separate two ensembles, and you can try to use that feature within enhanced sampling approach. But I would not consider MSM as particularly supervised, but it does need some feature as inputs, and there are some lag time we need to tune.
Okay, perfect. I have one question of my own. What about if you have different cryptic pocket on one protein? How will you adapt the method to make sure that you can kind of find all of them? Because like in terms of space, then you have cases where one is open and then the other one is closed, and then maybe two of them are open. Like how for example, taking this example, how will you change the Markov chain to kind of account for such case?
So the way we try to do now, it's a slightly expensive approach, but we know that protein goes back and forth between closed and open states. So, and we know what is our cryptic inhibitor. So what we did, we put our cryptic ligand in a closed state, and we ran an adaptive sampling approach, which tries to find new states during simulation, and it induced a change which shifted the equilibrium from closed to open state. So we had from the APO the idea of an open and closed state that I'm showing. We put a ligand which is bound to, which should be bound to an open state, but we put it in the closed state because we believe the ligand will induce the change. Then we ran adaptive sampling, which just is a kind of objective function which forces the protein to go to a different state, sample more states. And after running a few rounds of this adaptive sampling, the closed state become open state. So now we know that this kind of scaffold induces this kind of change. So because we already have the idea of the ligand, we can now design analogs or use generative modeling and other approaches on the ligand to optimize it. Does it answer the question?
Yes, it does. If a follow-up will be, then what happens if you don't have the ligand to start with?
Yes, what properties the ligand has?
If you don't have the ligand to do the simulation to go from the closed from the closed to open set.
Yeah.
How do you manage the sampling then?
So if you don't have any kind of intuition of the ligand, what we have seen in by analyzing some of the cryptic pockets, it's very hydrophobic in nature. So in most cases, so we try to give that part is heavily packed by a benzene or other, or like a chloride benzene, like kind of like a hydrophobic part of the, so which you can rationalize by doing a database search or like kind of a generative modeling, giving a constraint that it has to be hydrophobic in nature, because the cryptic pocket usually so far is more tending towards more hydrophobic.
Okay, I see. Thank you. Question in the chat. Any thought on using your approach for some target for small molecule discovery for cancer disease, for example? Also, could you share the small molecule cryptic pocket work? I think I will share the link, but you can take the first question.
Yeah. So to answer the first question, we are working on a cryptic pocket idea for a cancer target. I cannot disclose the name. And there has been a plenty of, recently there has been a bunch of good work on targeting cryptic pocket by small molecules, which I can share. There are a few, if you just search in the literature, there are definitely good works from targeting cryptic pockets in small molecules. But yes, cancer is one of the cases where the selectivity will be a big problem, and targeting cryptic pocket has the chance of addressing that. So we're actively working on that area.
Perfect. Any other questions? If not, we call this one a short one. And thank you again so much, Rakat, for the good presentation and answering all the questions.
Hi, I have a quick question for Somendranath. Somendranath, first of all, excellent talk. Loved it. So you mentioned that some other work following your work was like a validation of your work because they used and they arrived at the same pocket. So I was referring, can you share the URL for that paper or the name of that paper in the chat?
Yeah, absolutely. Give me a second. I will do that.
And just out of curiosity, are you Professor Bowman's student?
I worked with Greg when I was a postdoc, yeah.
Okay. Yeah, I'm a huge fan of, so good to see somebody from his group. Yeah. Thank you.
Yeah, thanks.